All right, young folk. Today we're going to be talking about DNA. Um, this video that's about to pop up, not the one that I'm talking in. I repeat, if you come up to me with just two notes over this whole PowerPoint, over this whole like 20-minute video, we might fist fight. I'm just kidding. I wouldn't do that. But you won't get your points. So you're going to watch this video that's about to play. You're going to take two notes over it. After that video that pops up on the screen is done playing, you're going to continue to take notes throughout the rest of the video of Mr. Moore talking, okay? So make sure you're prepared. Make sure you're focused on the fact that you're going to take two notes from the video that is about to play, not the video that Mr. Moore is talking in. All right. So make sure you have those two notes, and we're going to move on. Remember, after those two notes, continue to take notes. So what is DNA? Well, let's talk about it a little bit.
Hi, I'm Emerald Robinson, and in this What Is video, we're going to examine the so-called stuff of life, DNA. DNA is a nucleic acid that carries all the genetic material necessary for cells to develop and function. The letters DNA stand for deoxyribonucleic acid. DNA floats freely in the cytoplasm of bacterial cells and is found in the nucleus of plant, animal, and fungal cells. DNA is made of subunits called nucleotides. Each nucleotide has a phosphorus group, a sugar, and a special nitrogen-containing molecule called a base. There are 4 of these bases in DNA: adenine, guanine, cytosine, and thymine. Cytosine and guanine combine, just as adenine and thymine combine to form units called base pairs. When the nucleotide subunits are put together, They form a structure that looks like a twisted ladder called a double helix. The phosphorus and sugar molecules make the rails of the ladder, and the base pairs make the rungs. The double helix model was suggested by scientists James Watson and Francis Crick in 1953, and it earned them a Nobel Prize. DNA acts as a blueprint for the cell because it contains the codes that a cell needs to grow and function. These codes come from specific segments of DNA called genes. DNA and the genes that make it up are passed from parent to offspring, so the information carried on your genes determines who you are. The result of a gene being expressed is often a protein. These proteins ultimately determine traits like blood type, hair or fur color, and likelihood of getting certain diseases. DNA is unique because it replicates itself, making it essential for cellular life. When cells divide, each new cell needs an exact copy of the DNA present in the old cell. This copy genetic material allows all modern living things to function, grow, and reproduce.
For thousands of how-to and advice videos on any topic, visit monkeysee.com.
All right, so DNA stands for deoxyribonucleic acid. Again, I'm going to say that, deoxyribonucleic acid. We have absolutely seen this word probably about 45 to 50 times already this year. And I know for a fact that you've seen it multiple other times in your past science classes. So a DNA molecule has the shape of a double helix. So basically what you want to imagine is a ladder that we grab the ends of on both sides and just twist. All right? Each strand of DNA helix is a chain of nucleotides. So that means it's going to have deoxyribose, and cytosine really is it's just one of the four. So it could have adenine, it could have thymine, and it could have guanine as well as cytosine. But we're using cytosine in this place because that's what's on your test in the question. Don't tell anybody I told you. Okay? And phosphate. So basically you're going to get a sugar, a base, and a phosphate group. Your sugar is the ribose, the deoxyribose, and your base is adenine, guanine, or cytosine. All right? DNA information. Let's see what
everything you do changes your DNA, and then that can change the DNA of your children or future children. I'm not here to pressure you into anything. Anthony here for D News, and you've probably heard of epigenetics. This is the idea that while the structure of your DNA doesn't change, parts of it can be turned on and off through chemical changes in your body. It's called DNA expression, and it has a lot to do with your behavior and lifestyle. So smoking and eating too much can make the genes for obesity in your body express themselves strongly. Psychological trauma and highly stressful events can affect the genes that control the brain receptor for oxytocin, which is this hormone associated with love and trust. People who sleep poorly for just a week can show changes in 700 genes. Now, the good news is that the opposite is also true. So leading a healthy life can cause your body to turn on beneficial genes. You can also reduce the genetic risk for diseases that are hereditary in your family. There's even an idea that you pass those changes on to your children. It's called epigenetic inheritance, and we used to think that its effects were minimal, that genes would reset their on-off switches between generations. But a small study published this week by the Proceedings of the National Academy of Sciences showed that it may play a huge part in a child's development. So researchers tracked 20 women, all mothers who had had gastric bypass surgery and lost around 100 pounds. All of them had children before and after the surgery, not for the study, but because they wanted a family. When the team compared the children born after the mother's surgery to those born before, there were about 5,698 genes that were expressed differently, and most of them were genes that are important for heart health and metabolism. And when the doctors tested the kids with the changed genes, they had healthier insulin levels, blood pressures, and their weights were better than the other kids. Epigenetics might even control love. Love, man! There are these prairie voles that mate for life, they build nests together, they get studied a lot because their relationships look a lot like human ones. In a study published by Nature Neuroscience, researchers took two voles that had been around each other for 6 hours but hadn't mated. and they injected them with this drug that was supposed to stop methylation of the genes that controlled oxytocin and vasopressin, which are the big love and trust hormones, right? Total love connection. The voles fell for each other, and when their brains were compared to a couple of voles that made it on their own, the oxytocin and vasopressin receptors looked similar. Love causes the change causes love. Something interesting. If the scientists injected two voles who didn't know each other with the drug, the love connection didn't happen. Which brings up the big question with epigenetics. How much control does it really have over us? Those voles needed some sort of pre-existing bond, even if their genes were telling them to build a tiny vole love nest and have 2.5 vole children. You might be able to restrict or strengthen the expression of a gene somewhat, but can it really fundamentally change you? What do you think? Let me know down below and subscribe for more D News.
All right. So you got a little bit of information on DNA. We compare this to a book because it stores information, it copies information, and it transmits information. That is what DNA does. It's also important that there's accurate transmission of this information in the sex cells. Why is it the most important in the sex cells? Because when the sex cells come together, sperm and egg, they come together. That creates a baby. If the baby doesn't have the correct DNA or genes, it's not going to develop correctly. This can cause diseases such as Klinefelter syndrome, Down syndrome. Can also cause like 6 fingers to be on one hand, 6 toes, 1 eye, things of that nature. Now, if this happens, if DNA gets damaged or goes missing, then the information that was there gets lost. Cool thing about your DNA, though, is it can repair itself. The problem is if we lose a piece of genetic information, your DNA just guesses. It doesn't actually know exactly what goes there. It can just guess at what goes there if both pieces get lost. But we can talk about that a little more when we need to in later science classes. DNA damage.
Mistakes in base pairing occur during replication of DNA. Mistakes in base pairing occur during replication of DNA.
Okay, good.
To certain chemicals. Such damage can result in alteration of the genetic information. The repair mechanism recognizes and removes incorrect or damaged nucleotides. The undamaged strand serves as a template for replacement with appropriate intact nucleotides.
All right, well, that was a short video. This can cause the cell... Okay, this is annoying. Hang on. Patience with me, young children. My bad. I know. I messed up. It's on me. But we're going to just go with it. I'm going to pause this video so we don't have to listen to it again. Anyway, this can cause the cell to not function correctly. It can store damage... I'm so sorry. That threw me off. Now I'm a little off my game. So let me just take a minute. And we're back. Storing damage... Cell cannot make the material, and it actually causes the cell to not be able to function. Copying damage, you guys can read. If we copy damage, then obviously it's going to do the same thing as if we store damage. It's not going to actually work. And in some cases, the cell will be actually die, like legitimately die, because it just can't function without the proper DNA. So. Yeah, so if the offspring don't get all the information, then they obviously can't participate in the important things of creating what needs to be created. So if we have damaged DNA, the offspring won't get the information. That information can actually be necessary for life. Like, let's say a gene goes missing that's supposed to help you with your heart, to form your heart, and that goes missing, then you won't form your heart, right? So then you could not have a heart, and then you wouldn't be able to live. That'd be sad. Mr. Moore's off his game a little bit tonight. I don't know why. Probably because the TV's on in the background. But that's okay. Just write down what's in yellow, and if you have questions, I can answer them because the TV won't be on in class. Well, DNA replication and copying. This video is so good. I'm about to give up on my videos. So anyway, this results in 2 DNA molecules, each with 1 new strand and 1 original strand. That is actually extremely important to remember because we have 1 new strand and 1 original strand. So what happens is the DNA actually unzips, like it unwinds and then unzips like a zipper, and then molecules come in, and we know we have base pairing, right? Well, once the base pairs, or once the DNA, the molecules come in, they pair up correctly, and they connect to one another, and they create 2 strands, with one side being a new strand and the other side being the original strand. For replication to happen, hydrogen bonds between the base pairs must be broken. That's where we get it unzipped. So in between each base pair, there's a bond of hydrogen molecules, and they're just not very strong, so that's where we unzip. Once these bonds are broken, it unwinds or unzips, and then the 2 strands are held together by the hydrogen bonds between the DNA. So those hydrogen bonds actually reform to create something new, or to create new DNA molecules. So base pairs, I guess this is on me because we didn't get to base pairs prior to, but let's see what this video, if this video wants to participate.
Guanine, cytosine, and thymine. Not exactly words that slip off the tip of your tongue. But without them, you wouldn't have a tongue. But
True.
Let's move on. These nitrogenous bases create the sequence that we think of as genetic code. Remembering how the bases form base pairs will be important as you learn more about DNA. Replication, transcription, etc. But we're getting ahead of ourselves. As a shortcut, biologists often refer to the nucleotides in DNA by just the first letters of their base names: A, G, C, and T. Sort of like rock stars that are known by a single name. In 1949, Erwin Chargaff discovered that when sorting out DNA, the relative amounts of A and T were almost always equal. And the same is true for G and C. In 1953, Watson and Crick figured out, with help from lots of friends, that hydrogen bonds could create a nearly perfect fit between nitrogenous bases along the center of 2 strands of DNA. In fact, these hydrogen bonds form between only certain base pairs, and remembering these base pairs is key to our understanding how the code in DNA is passed on. From Chargaff's data, it's not hard to figure out that guanine bonds with cytosine and adenine bonds with thymine. But how can we help ourselves remember the pairs if, say, we have a really important test we need to pass? There are only 2 possible pairs. A pairs with T, and C pairs with G. If we can remember just one pair, we can figure out the other one. Here's a trick I use to remember the pairs. If you list out the 4 letters, A, G, C, and T, and take a look at the letters, you see that G and C are 2 letters with similar shapes. This similarity is how I remember that G and C form one of the base pairs. Now that I've matched G to C, that leaves A and T. So I know that A and T go together to form a base pair. So this simple trick, noting the similar shape of the letters G and C, is how I remember how the 4 nitrogenous bases match up to form base pairs.
So I hope you have a really, really, really good imagination, because quite frankly, I did not realize that video was not playing the way it should. It tricked me. So I apologize. I will take the time to draw this on the board at the end of the class period so that everybody gets to this point. I'm pretty sure, if I'm not incorrect, this is close to the last slide. I'm not 100% sure, but it's either 1 or 2 slides away from the end. So I'll draw that on the board. But base pairs are the building blocks of DNA. A equals adenine, T equals thymine, G equals guanine, and C equals cytosine. Adenine always forms a pair with thymine, and guanine always forms a pair with cytosine. So, for example, a DNA strand that has the bases CTA, GGT will produce a strand with the bases GAT, CCA. So G and C always go together. T and A always go together. So I'm going to take a moment with my sloppy handwriting, which is going to be even worse on this little PowerPoint. But I want you to write the match up with, we've got an A. That's an A, yeah, I know. We got a T, C, and G. And then we'll go T, G. All right? So what I want you to do is I want you to give me the other side of that. What's the other side? If you follow these rules here to the left, I want you to write in your notes ATCG, and then right underneath the ATCG, I want you to write what would match up on the opposite side, or what we call the complementary pair. Okay? So you're going to write that down in your notes. You're going to write it out, and then you're going to show it to me. I'm going to say, let's say the first 3 people that show it to me will get extra credit. So the first 3 people that show it to me and are correct will get extra credit. And that's the end of the slideshow. So look at that. Perfect timing. So whoever gets done first is superhero, and they get extra credit. Nobody else does because, well, the first 3 do. But once you're done, come show me your notes. First 3 people done with that extra piece will get the extra credit. If you have any questions, feel free to ask. Stop your video. Call me over. I'm not doing anything. I'm just sitting here waiting for people to ask me questions. So let me know. All right, you guys have a good rest of the day. Get ready for your next assignment.